Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this little notescast, we're going to be talking about the cell membrane. You're also going to see this written down as the plasma membrane, and you can use those terms interchangeably. We'll start talking about the structure of the cell membrane and use that as an introduction into the function of the cell membrane. And here in these scanning electron micrographs, especially in this one here, you can see the overall basic structure of the cell membrane is a bilayer. More specifically, it's a lipid bilayer, but we'll talk about the lipids there in a second. But here you can see in this other picture how one side of the cell membrane on this side is clearly different, has a different characteristic than the other side of the cell membrane. And that's the basic job, is to maintain that difference, the different environment inside the cell than outside of the cell. Here's a computerized rendition of what the phospholipid bilayer looks like. And here you can see a little better the individual phosphates of the phospholipids and their leggy hydrocarbon chains here in the middle. There are also interspersed amongst those legs some cholesterol molecules, and those help keep the legs kind of tangled so that they maintain the cell membrane fluidity. Now, when we start talking about the cell membrane itself and the phospholipids, it's always good to start with a discussion of soap, exactly what soap is. Now, what we do is to make soap, we take lipids and heat them up in the presence of a strong base. In this case, sodium hydroxide works very well. And what that'll do is it'll break the fatty acids off of the glycerol molecule. Remember, a lipid is made up of, at least the triacylglycerides that we talk about are made up of a glycerol and 3 fatty acids. So we'll break the fatty acids off and then stick a big sodium ion up on the top. And what that does is it makes a special kind of a molecule that has on one side of it parts that likes water and then on the other side of it part that doesn't like water. And this is how soap actually works. You get a little bit of grease in water, and if you put some soap in there, the legs of the fatty acid go into the soap, and then that surrounds it with the heads that like to be in water, and then that way we can dissolve that little blob of grease or of lipid in the water. And this happens spontaneously. It's just what soap molecules do when they interact with hydrophilic and hydrophobic substances in the water. Now we can compare the action and the structure of a soap molecule to the phospholipid that makes up the cell membrane. And here we have the head of the phospholipid. It's a big phosphate group. It's charged just like the sodium ion, and that makes it mix in water or be what we call hydrophilic, water-loving. The tails, the hydrocarbon parts of the fatty acids, are hydrophobic. They don't like to be in water at all. They don't mix well with water. They're what we call nonpolar. They can hang out with other tails, but they don't like to be in water. And since the outside environment, cells are in a watery environment, and cells themselves, the cytoplasm, is mostly water, and so that's a water environment. Just like soap molecules don't bond to one another, the individual phospholipids don't bond to one another, and it's this hydrophilic, hydrophobic interactions that keep them in place. And just like soap molecules, we can take, just like you can take one soap bubble and make it into 2 bubbles, we can take one cell and pinch into 2 cells. We can make vacuoles similarly. That makes it a dynamic structure. A mosaic is art that is made of different colored tiles or different pieces of glass or stones or something that are cemented in place in an artistic arrangement. And we can talk about the structure of the cell membrane as being kind of similar in that there are all these proteins that are not really embedded, but they're surrounded by these phospholipids in that phospholipid bilayer. And so what we end up with are kind of like the tiles of the mosaic are these proteins that instead of being cemented in place are able to flow with the fluidity of the cell membrane. And so that's why we call it the fluid mosaic model. Let's talk about some of those tiles, some of those proteins and other things that are part of the cell membrane. Stuck to some of the proteins and to some of the lipids of the cell membrane are these carbohydrate chains, and they have particular shapes. They look like trees. but they have particular shapes, and each individual, each person has their own shaped identifiers, their carbohydrate trees, and these kind of act like Braille name tags. They allow cells to talk to one another and to identify one another. How they feel one another's carbohydrate trees are going to be the proteins that are also on the surface of the cell membrane, and many of those are going to form these transport tubes that allow materials to go through the cell membrane. We'll talk about those in a second. But others are called receptor molecules, and they act as like binding sites for, among other things, carbohydrate chains, these identifiers from other cells, but that will also interact with hormones and other communication, cellular communication molecules. So these receptor molecules become really, really important when it comes to the function of the cell membrane. These receptor molecules are proteins, and we know that a key to a protein's function is its shape, and that's going to be the case with these receptor molecules, just like it was the case with enzymes having a particular shape to match with a particular substrate. These receptor molecules are going to be key to the transport functions of cell membranes and other functions of the cell as well. It's going to allow the cells, like immune cells, to be able to tell if other cells belong there or not, if they're self or non-self, or if they're abnormal, by kind of feeling the shape of those carbohydrate trees. If they fit into particular receptor molecules, then the immune cells know they belong there or don't belong there, and what to do with those cells. It's also going to allow cells to tell if molecules are useful for ingestion or not, if they're food molecules. It's also going to allow cells in us big clunky multicellular organisms to talk to one another. So one cell can release a hormone or other signaling compound that can get picked up by the receptor molecules of other cells and cause a change in that cell, and that's how our cells talk to one another. We have to be ready for lots of different artist renditions of cell membranes. So here's another one. And here's another one, but they all have kind of the same idea. Here's these two layers of phospholipids with these big proteins embedded in there. Some of them are transport proteins, and some of them have particular shapes like for the binding sites. So they all have the same idea. The fluid mosaic model lends itself to the transport functions of the cell membrane, that is, getting material in and out of the cell, allowing some material to go through while blocking others and maintaining that different environment inside and out. Now, like I said, some material is just going to be able to pass through what we call diffusion. The cell membrane isn't going to put up much resistance to, say, water molecules, as it has these aquaporin transport channels that allows the water to go right through. So if there's more water on one side of the cell membrane, it's going to flow the other direction. And so the word we use for this is semi-permeable. That is, some material is allowed through while others are blocked. And the semi-permeability, like I said, maintains a different environment inside the cell than outside the cell. But still, other material has to get through if it can't just diffuse. And so there's a way to pump that material through. And that's what some of those transport proteins are going to allow for. Other material can fool its way in. We really don't want it to get in, but if it has the right shape and can get through because it's shaped properly to get through a receptor molecule, then it's going to be able to get into the cell as well. The main function then of the plasma membrane is to maintain that constant internal environment, that constant concentration of chemicals on the inside of the cell, even though the outside of the cell is going to have different concentration of chemicals and it's not going to be stable. So it's going to regulate homeostasis. It's going to do this by affecting which molecules are able to cross and when they're able to cross, and if they're not going to cross at all. Now, how they're going to do this is by controlling what's called active and passive transport. Some material is going to pass through the plasma membrane through passive diffusion. Some stuff is going to have to be actively pumped through the cell membrane, and this is where some of those receptor molecules are going to come in through the process of receptor-mediated endocytosis. This comes in a couple different varieties, which we call phagocytosis, where the cell eats big chunky bits, as well as pinocytosis, where it kind of gathers up bits of the external environment and gets the dissolved materials as well, kind of like cell drinking.
Well, let's stop there for now. We'll talk about active and passive transport in the next video anyway. Thanks again for checking in to Mr. Ulrich's landofbiology.com. Again, I am Mr. Ulrich, and if you have any questions, feel free to drop me an email or send me questions in the comment section below. Other than that, we'll see you in class.